everyone. Welcome to the next lecture, which is about pheromones, a very interesting topic with some fascinating things happening in it, as you will see. So this is perhaps the most well-known example of chemical ecology. And pheromone is defined as a chemical signal which is released to the outside of the body of the producer that affects the physiology or behavior of a receiving individual, and this is the critical thing, of the same species. So it's a within species signal, and the name comes from the Greek pherin, to carry, and hormone, to excite. So it carries excitement, and the best known of the pheromones are the sex pheromones, and so this is something which is often of a very great excitement when the individual of the opposite sex encounters this signal, because it means that the mate is there, and so it's a very powerful signal. And there are actually some other types of pheromones too, which I will also show you, but the best known ones are the sex pheromones. These pheromones, they're produced from exocrine glands. So it's like a hormone is produced from the endocrine gland, which goes inside the body. The exocrine gland is for a secretion which goes to the outside of the body, because it needs to go outside so that it can get to another individual of the same species, as we will see. So pheromones, they were actually have some history to their discovery, and there's a very nice story of this French naturalist called Jean-Henri Fabre, and in the 1870s, it goes back to this. He was very pleased when he was in his laboratory study area, and a great peacock moth emerged from the cocoon that he'd been hatching it from on his table, and so this is the story. And he put her under a wire gauze bell jar, so to have some aeration, but so she couldn't escape, and left her to let the wings dry. And then about 9 o'clock that evening, to Fabre's pleasure, turned to amazement, as dozens of great male great peacocks with striking eye spots on the wings, and as much as 6 inches across, floated in through the open doors and windows of the house. So it's the males of the same species are arriving, and he's wondering, well, how did that happen? Because how did they find her? And so this is the quotation of what he says. And coming from every direction and apprised I know not where, Fabre wrote, "Here are 40 lovers eager to pay their respects to the marriageable bride born that morning amid the mysteries of my study." So he was a bit puzzled by this, about how did they find her. And over the following week, he caught more than 150 males who were coming along, and no matter where in the house he moved the female, the male moths made directly for her. So what was drawing them in, he wondered. And so this was when the idea of that a pheromone could exist was first being thought about. So it's rather a wonderful thing, really. And in this next slide, you can see some of this in action. So there's a video clip I found on YouTube, and this is a hemileucea moth. And if we just play this, you can see what happens. So it's a bit similar to what Fabre observed. So you can observe it too. So there's this moth there, and then they're putting it in this net. There's the female there, and it's in this butterfly net. now the males are coming along. So a similar thing to what Fabre observed. You can see it there in that video clip. And so that's the sex pheromone. And to recap, the pheromone, it's a chemical signal conveying information between members of the same species. And so the sex pheromone is used to call the opposite sex in mate finding behaviour. But there's also other sorts of pheromones, so which can also be important for the fitness of these insects and other organisms that use them. And there's also alarm pheromones that exist, and so these allow them to warn each other of danger. For example, when a predator is attacking, there's aggregation pheromones. So those attract both sexes of the same species, and this might be used to overcome host defences if they can do that better when there's a large number of them, or to form a group against predators, so that it'll be more off-putting to a predator perhaps to see a large number of them, and it might not attack them. And or it can also serve the purpose to have an aggregation of them for mating purposes, so they all coming together. It's not actually a sex pheromone, but it still helps them to aggregate for mating purposes. So there's a number of possible reasons why you might have an aggregation pheromone, and so these also exist. Then in foraging behaviour, particularly with social insects, I'll show you with an ant in a minute. But these help them to find food sources. So when another individual of the colony has already found some food, then they can sort of lay down a pheromone trail to help other individuals to find that food resource. And then finally, there can be host marking or spacing pheromones. So if there's only enough resources for a limited number to survive, this might happen, for example, with egg-laying behaviour. Then it'll lay down a pheromone to warn other individuals of the same species that an egg has already been laid in that spot, or something like that. So this avoids multiple parasitism if there's only enough resources for one individual to survive. So that's another type of pheromone. There can be all sorts of different spacing pheromones as well. So they can be another useful thing. And then oh yes, finally there's one more. There's a identification pheromone, and so that can be used by social insects to see if there's another individual of the same colony. So it's got this thing called the home odor, which they can use to recognise if there's other individuals who are in the same gang as them. So that way they can tell the different colonies apart. So there's different sorts of pheromones. Oh yeah, so there's something popping up there with a alarm pheromone and an insect going on the trail. But I'll show you that in a bit more detail actually. Just coming up in a bit. But going back to the sex pheromones, the sex pheromones are best known and best studied. Function is gender attraction, so it's most frequently it's the females calling to the males. So it's almost always like that with insects. I'm not sure exactly why, but that just seems to be what has evolved, and so it's the female that will be giving off the smell, and it will be the male that's got the job of finding it and flying towards the female. But there are occasional examples of males calling to females, but this is much rarer. But it does exist. There are some species which have it the other way around. And then the quantities of pheromones, because they're species specific signals, they don't need a large amount of it. It's to do with the particular chemical structure, and so it just needs enough of it for it to be able to detect it. And so there's often very small quantities of sex pheromone being produced, and it'll be generally in the order of nanograms, or sometimes even picograms, especially for the midge pheromones. They tend to be produced in really minute quantities, which means that only a really tiny amount of it can have a really powerful effect. So it's quite amazing stuff. But where they have them production in small quantities, it does make it challenging for us chemical ecologists when we're trying to identify what they are, because then we've got to work with really tiny amounts of material, and that makes it quite hard with the chemistry for identifying what on earth it is. So that's those are the and if you remember on the opening slide, I had this picture of a silk moth, and this was actually the first insect that the pheromone was identified from, and it has these rather remarkable antennae on the males. If you look at it, you can see it's got these hairs extending off the main antenna, which give it increased surface area. And this was the first insect that a sex pheromone was identified from. It was done by Butenelt in 1959, and they actually picked a difficult one, because the sharp the sharp thinking of you may have already guessed this, but the reason why it's got these big branches on its antennae and for increasing the surface area is so that it can be more sensitive for picking up the tiny amounts of pheromone. So the sick moths, they don't actually produce that much pheromone, so it was really quite a difficult job to do, and they had to extract pheromone from thousands of silk moths to get enough material to identify what it is. And there's really high sensitivity in this, so the male silk moth can respond to the female produced sex pheromone behaviourally when the antenna detects only one molecule per second. So that's pretty phenomenal sensitivity. It's actually a lot better than the chemical analysis machinery that the chemists have, and so they needed to get big quantities of insects to compensate for that, especially going back to the technology that's available in 1959. Things have improved since then, and you get better sensitivity, and you can identify things in smaller amounts. But it's quite remarkable that was the first one to be identified, and it causes this very predictable response in the male. So as soon as they smell that stuff, they get excited. They start fanning their wings, start approaching the female and everything. So it's eliciting these mate approach responses, as you can see in the picture there. And there's extreme sensitivity in the olfactory system. So it's really amazing, actually, how sensitive these insects can be. Another example here is from Bill Hansen's group. This is when he was still in Sweden. He's now in Germany. But they were looking at this moth, and again, a lot of the chemical studies are done in moths, and they found that there was a heartbeat response was triggered by less than 6 molecules of the most efficient odors hitting the antenna of the insect. So that's really phenomenal sensitivity again there. And if I'm not wrong, I think this wasn't actually or this can actually occur even with host plant materials as well. I think they tried a number of different things. So they were using sex pheromones, but there's also host plant volatiles where they can get really high sensitivity too. So this really shows how sensitive they are, because we're talking about just 6 molecules causing behavioural response, and I think that's pretty amazing. So the pheromones, you can get them from all different classes of chemicals. So these different functional groups of chemicals, and just depends on what happens to have evolved. I mean, different groups of insects will have evolved responsiveness to different molecules, and so you can have alcohols, you can have aldehydes, carboxylic acids, and you can see there on the slide there's examples of insects that are responding to sex pheromones from all these different classes of chemicals. So it just depends what has evolved, and there's a great deal of selectivity, because these are species specific signals, so you don't want to be attracted to a different species that you can't mate with. So there's very strong selection pressure for them to be able to find the compatible mates that they can mate with of the same species, and to be able to differentiate from each other that way. In the early days of pheromone research, they might have thought, oh, there's just one sex pheromone, but when it was studied in more detail, then people found out that quite often there's actually a mixture of different pheromone compounds, and quite often there'll be one component which is more abundant, but then there'll also be some other sort of minor components, and these can increase the potency of the pheromone. So it'll have a particular ratio of different chemicals. Very often, I mean, sometimes you'll be able to get the whole response with just one chemical, but in other species, then you might need to have a mixture of pheromone chemicals, and there's these different structures of the different ones that you can see here. Some examples here, and the tortricid and parallelid moths, they've got this tetradecane acetates. There's hexadecanal and tetradecanal in these heliothiene moths. There's different ones in different species, and these have been identified. A lot of these studies are focusing on pest species, so they're identifying the pheromones because they're hoping that these might be useful as interventions for managing pest species of insects. Because if you can catch them with a pheromone, then maybe you can manage the pest. But that's the topic of another talk, so I'll go into more detail about that later. But you can see that a lot of these studies, they've focused on insects that are economically important as agricultural pests or as vectors of human diseases. Something else to notice about these pheromones, and another intriguing thing, is that that you can get different response to the different enantiomers. So I don't know if you remember from your chemistry what an enantiomer is, but it's the same chemical structure, the same arrangement and everything, but it's the mirror image. So an analogy for this could be that you've got your two hands, your left hand and your right hand, and the fingers they're all arranged in the same way, but they're mirror images, so you can't actually superimpose them because they're mirror images. And it's the same with these chemicals. So you can get an S form and an R form, and they're actually different molecules because you can't superimpose them. They are actually different, even though they've got the same they've got the same structure. It's just a mirror image, and the insects are so sensitive that sometimes the opposite enantiomer can actually have an inhibitory effect. So it no longer responds to the sex pheromone if it has sometimes even small amounts of the other enantiomer present. So sometimes there's an enantiomer specific response, and it needs to have not only that chemical, but it needs to have the right enantiomer of it. So it's quite a sensitive thing, and it has a receptor for it, which is highly selective, only responding to one enantiomer. There's other examples where they'll respond to both enantiomers, or there won't be any inhibition. But it depends on which species you're looking at, really. But sometimes the enantiomer needs to be spot on, or otherwise you're not going to get the proper response. So here's some examples of this. So the first, the top line there, you've got the Dendroctonus spark beetle there, sex pheromone. It's called frontalin, and you've got this this 1S 5R active enantiomer of it. But then there's another one which isn't an inhibitor, but it doesn't respond to it. You won't get a sexual response to that compound because it's not the right thing. And then middle line, we've got the gypsy moth pheromone. There's dispar lure, and you've got the 7R 8S configuration is active. The other one, 7S 8R, is actually inhibitory. So if you have some of that there, it's actually going to switch off the response to the active one. So if you've got some contamination with that, your sex pheromone mixture isn't going to work anymore. And it's quite important with some of the applied work that people try to do with this to see if they can use it for trapping the insects. And then there's another one example of on the bottom line there, cigarette beetle. It's got an 4S, 6S, 7S, which is active, and 4S, 6S, 7R, which is inhibitory. So some nice examples there showing how that can be and how it varies from species to species. And this shows about the blends. So the closely related species of moths, they'll sometimes have similar molecules they're using for mate recognition, but they can differentiate because it's going to be in some different combinations and different ratios. This is also the case actually when insects are responding to host location cues, but again that's a topic for another talk. I'll talk about that separately. But as well as having the chemical, it's also having the ratio of different compounds and the particular combination that needs to be the right one in order to get a full response or any response. So it's and they can distinguish between the different species. So even though there'll be some overlap, and there'll be some pheromones, some pheromone chemicals which are in common. There'll be some other ones which they also require, or it needs to be present in a different amount, so they can still distinguish each other that way. Okay, so I think we've said enough now about sex pheromones. Let's have something different, and this is aphid alarm pheromone. And so when the aphid is attacked by the predator, then there's a lacewing larva there in that slide. It takes a bite out of it. I mean that aphid has had it, but these aphids, they exist in groups and they're actually clones of each other due to their rather special reproductive mode. They have asexual reproduction, and so the summer forms of the aphids, they're actually clones of each other. But they warn their neighbouring aphids. So although that one's had it and that was going to get eaten, you can see there's a droplet coming out of the cornicle at the rear end, and it's not just that sticky defensive secretion, but there's also a volatile in there, so that it's producing a smell. And you can see the chemical structure of that smell up there as well. It's this compound E-beta-farnesene, and this is the aphid alarm pheromone. So when an aphid detects the alarm pheromone, you see there's a drop of it there, a solution of it being put on that leaf. Normally aphids are very sluggish and they don't move around very much. But as soon as they smell that smell, they're off, because they associate that with predator and with danger. And so when they smell the alarm pheromone and they get a sudden whiff of that, then they know that they're in danger. And so they normally have their stylets plugged into the leaf, and they're not moving around very much. They're just sort of tapping the leaf and draining it of resources. But once they can smell this alarm pheromone, it's playing it again now, you can see it again, then they're up and they're off, and for an aphid this is really high speed running away. Normally they're just sitting still for hours on end. So this is fast movement for an aphid. They're all clearing off and moving away, and so that's the way they can warn each other of trail pheromone. So this is a colonial, social insects and colonies and their foraging success depends on how efficiently they can communicate to other individuals. Same problem about the resources. So they can tell other ones that they can find the food and bring it back to the nest. Workers for exploiting food resources more efficiently if they lay down a trail pheromone. And there's actually some nice history at Keele of identification of trail pheromones of ants. So there's quite famous chemical ecologist called David Morgan, he's at Keele, and he did quite a few studies of the trail pheromones of ants, and got this very nice publication where he's showing all these different trail pheromone structures that they've identified. So that's quite nice that those pheromone studies were done at Keele. And my colleague William Kirk has also been involved in identification of thrips aggregation pheromones. And so just to conclude with, pheromones, these are species-specific signals that are used for signaling to members of the same species. They can be active in really tiny amounts, so you don't need much of it. It's the chemical structure that is the critical thing, and sometimes the blended ratio of different components. But it can work in really small amounts. The ability to respond to these pheromones, it improves the mating success of the insects, and so there's very strong selection pressure for them to be able to do this, because it's a critical thing for their evolutionary fitness. So that's how these rather amazing things have evolved. And there've been efforts to try and exploit these for pest management purposes, but I'll talk about that in a separate talk. So thanks very much for listening, guys. If you've got any questions, we're going to hold a workshop where you can ask questions. So I look forward to seeing you in person there, where we can discuss this talk and any of the other talks as well. So thanks very much, and that's it for now. Bye bye from me.